Okay, so let's start chapter 10. This chapter talk about different research that paved the way to understand the genetics. Okay, so two separate line of research were going on during mid 1800s. Okay, one was by Mischer and another one was by Mendel. Mischer, he isolated nuclein, and Mendel, he demonstrated the basic principle or pattern of inheritance. Now, before this time, like when Mischer and Mendel was working on to find out that the hereditary basis of our life, we really need to know the background. So that time, before that time, actually, the people used to believe in blended pattern. Blended pattern means we did not have any idea about what is gene or chromosome or how actually offspring get the traits from the parent. We did not have any idea. People used to believe in blended pattern. Blended pattern represent um, basically it says, or people used to believe that um, offspring actually get both the traits from the parents, and as a result they get a mixed trait. Okay, so if just imagine it's a pink flower. Let's say it's a pink flower. Okay, so sorry, it's a if it is a red flower, and let's say it's a white flower. Okay, then and if we cross between these two plants, so one plant with red flower, the other plant with white flower, then always the offspring will be pink. Means the plant will produce pink flower. So basically the pink is intermediate between the red and white. So if the mom has blue eye and dad has a black eye, child is going to get eye with intermediate color between black and blue. So that's the blended pattern, okay, or blended theory of heredity. So people used to believe on that because we really did not have any idea about the existence of chromosomes or DNA or the gene. So Miser, what he did, he isolated this nuclein. That's a very phosphorus rich chemicals, and he isolated that chemicals from the nucleus of WBC, or white blood cells, leukocytes actually. So he isolated these phosphorus rich chemicals from the nucleus of a cell, so he named it as nuclein. The importance of that discovery was the presence of phosphorus inside the nucleus. So at least it gave us one idea or concept that there is something inside the nucleus that is rich in phosphorus. Okay, so that was very important. Now we know, like the DNA, it's rich in phosphorus, right? So that indicated the presence of uh, one hereditary material that that is actually rich in phosphorus. So That's why this research was very, very important. Now, Mendel, he demonstrated the basic pattern of inheritance, how actually the trait moves from parent to offspring. We came to know after Mendel's experiments on pea plant. So what he did, he chose pea plant, Pisum sativum. Okay, so that pea plant, there are so many reasons why he actually chose the pea plant. Few important characteristics feature of pea plant is it's a self fertilizing. Okay, self fertilizing plant means one individual plant actually produces both the male gamete and female gamete. Okay, so let's say if it is a flower or pea plant, okay, it has that stamen, okay, and it also has carpel. So that stamen that produce the male gamete means sperm, okay, and the carpel, inside this body, it produces ova or egg, and these two can fertilize together, okay, and it can make diploid gamete. So that's the self-fertilizing plant. And as a result, these are self-fertilizing. So the pea plant is actually, they can make true breeding organism. So true breeding organism means every time the plant produces the offspring that exactly look like parent. So the true breeding plant, they always produces offspring that look alike their parents. So these two are very important characteristics of the pea plants that actually attracted Mendel. There are more, like pea plant, it has lots of different visible characters, okay? Multiple different type of visible characters. So when I'm talking about characters, so just I'm going a little out of this chapter, and to let you know that when we talk about the character, so character basically, that's a visible trait that we can see, okay? Like let's say it's a height. That's a character. Now under one character, multiple different traits are there. So different traits. So traits, when we are talking about the height, so that's a character, and when it is trait, and I said that multiple different traits are there, we know that it could be tall, it could be short, it could be a medium height. So all these traits goes under one character, which is the height. Okay, so for the pea plant, so many visible characters were there, and those characters are very prominent. Like let's say one was height of the pea plant, one was the flower color, one was the shape of the pod, okay, pod of the the flower. Then so multiple different types of characters were there. That was the one of the reasons that Mendel actually chose that pea plant. Now what he did, so he took two true breeding pea plant, and remember I told true breeding means they always produce offsprings like the parent. So when it is true breeding plant means when if the parent is tall, always the offspring will be tall. If the parent, so it is parent is tall, so offspring will be tall. If parents are short, then always the offspring will be short. Okay, so that's the true breeding plant. So he chose one character. Okay, so many. He actually chose 7 different characters, but for each experiment he chose one character. So let's say the character is height. So he actually cross-fertilized between 2 different traits, 2 different plants with different traits. So what he did, he chose one tall plant and he cross-fertilized that plant with a short plant. So when I'm talking about the cross-fertilization means, remember I told at the beginning that these pea plants, they're self-fertilizing plant. It means within the flower, each plant, they carry both they are capable of producing both the male gamete and female gamete. Okay, and this male gamete can fertilize the female gamete and they can produce the fruit. Okay, so but what Mendel did, instead of allowing this plant to self-fertilize, what he did, he actually take the sperm from one plant and he actually spread or somehow inoculate that sperm with the eggs of other plant. Okay, so that's the cross fertilization, and it could be female, it could be male. So anything, like from short plant he can take either egg or sperm. From tall plant he could choose the sperm or ova. So anyway, that's a cross fertilization. So basically over here during cross fertilization, the male gametes and female gametes come from the different individual. It's not like self-fertilization, fertilizing plant. So once he did that, surprisingly what happened, all the plants became tall. But remember that time people used to believe in blended theory, blended pattern. So when he crossed a tall plant with a short plant, he expected the plant would be the medium height. Okay, but instead of that, all the plants became tall. And that actually surprised Mendel. Why all the plants are tall? And where did this trait go? Remember that time we really did not have idea about how traits move from parent to offspring. So that was another question, like where did that trait from the short plant go? So he did not have any idea. So all the plant, basically all 100% plant were tall. So unexpected results. So what he did, he allowed all these tall plant, like and he named this generation of tall plant as first filial generation, or F1. That's how actually he named all these plant. Okay, so what he did, now he allow this all F1 generation tall plant to do self-fertilization. Okay, and what he noticed that some of the plants were tall and there are few of the plant who were short. So basically this time that trait actually came back. Okay, and he got the ratio between, you know, the tall plant and short plant. The ratio was 3:1. Okay, so basically the tall plants were 3 times more than the short plant. Okay, so what happened? Okay. So now what he did, he took so now basically at this moment, till now he was looking at the phenotype, means the external features. Now he started analyzing the genetic arrangement or the genotype of all these plant. So then what he find out that so when plants, they produce the gametes, um, what happened, like let's say that this tall plant, and I hope you remember that all of our body cells, that's the diploid. Okay, so 2 in number. So for this tall plant, it must carry diploid number of chromosomes. So basically it should be double T, double T. And the short plant, it must be small t, small t. Why it's small t, it's not small s, or not just s's? I will talk that later. So what happened, he noticed that when the parent produce gametes are actually haploid. Okay, and that chromosome or that trait, so dependent traits are there. So when they produce gametes, the traits, different traits, actually segregate from each other. They get separated, and individual gamete independently can get the different traits. Okay, so over here, so both the genes, or both the traits were look alike. Okay, so all the gametes, 100% of the gametes got that uppercase T. Over here, exactly same way, both it was small, both small t, so all the gametes got small t traits. Now when this gamete fertilized this one, then together they make the plant who had both the traits, means the tall traits and the traits for the shortness. Okay. Now from this experiment, Mendel actually derived one law, which is the law of dominance. Okay, law of dominance and recessive action. So what he told, that when multiple genes are there, or multiple traits are there, one trait actually dominate the other traits. Okay, and he actually named that trait as dominant or dominant trait, and the other trait, that which one is, you know, is not dominant, so he named that one as recessive trait. Okay, so that's the law of dominance and recessive. So over here, that uppercase T is always dominant over the lowercase t. So that's the reason he concluded that because of the presence of dominant trait, that individual actually is going to get the phenotype or external feature that represent by that dominant trait. So over here at F1 generation, all the plant were tall because they got one trait from the from the tall parent and another trait from the short parent. So both the dominant and recessive traits were there. But so because of the presence of the dominant trait, all the plant, they became tall. Okay, so but over here, this time, like this generation was F2 generation, and that's how he named this generation when he got that 3:1 ratio. What happened? So this plant, when it has both the traits during the making of gametes, these two traits get separated from each other, and some of the gametes will carry the dominant traits, and some of the gametes will carry the recessive traits. So as a result, so one plant is that and another one is like that. So what will happen? So when these two, this gamete will fertilize this one, then this plant will be tall. When this plant will fertilize this one, this will be tall again. Okay. Now when these two, so this will be tall again. But when these two, the small t and small t, then that will make one short plant. So 3 tall plant and 1 short plant, and that's how he got this ratio, 3:1 ratio, and it's true for any characters. Okay, so that from this experiment, that's his second law, which is called the law of segregation. Let me change the color of the plant. So one was law of dominance, and the number 2 was law of segregation. So law of segregation talks about when parent produces gamete, the traits, they get separated from each other. Okay, the word segregation represents the separation. So the traits get separated from each other, and some of the gametes they get one trait, and some of the gamete gets other trait. That's the law of segregation, and that explains this result, 3:1. Okay. So over here in this experiment in your textbook, this was another characters that Mendel actually chose, the flower color. So he took violet and white. So 2 true breeders. Means remember that violet, this plant always will produce the plant with violet flower. This plant will always produce plant with white flowers. That's how that's why they are the true breeding plant. Now, during F1 generation or first filial generation, all the 100% of the plant will be with violet flower. Why? Because the violet or the purple, that's the dominant over the white one. Okay. Now, when these plants are going to self-fertilized, then what will happen? We are going to get 3 purple versus 1 white. Okay, that's the ratio we are going to get. Now, so that tells that this flower, that F1 generation, all the plants are hybrid. Hybrid means they carry the traits, one trait from this parent and another trait from this parent. So basically they carry both the dominant and recessive trait. So that's why they are the hybrid. So if this is the purple flower, so let's say uppercase P and white. So that's the rule. Like when we work on the genotype, we represent genotype like that way, like dominant a dominant trait. We go by the letter or alphabet of the dominant trait, okay, and we represent dominant with the uppercase and the recessive with the lowercase. So if that's the P generation, means first parent generation, then F1 generation, what will happen? It will produce all purple, but they are not the true breeders. Okay, look over here, both the traits are same. Both the traits are same, and that's the reason they are the true breeders. But over here, in this case, so they are hybrid because these plants, F1 generation plants, they are carrying both the dominant traits and the recessive traits. Okay, now F2 generation, that's the law of segregation. These two traits get separated from each other during the formation of gametes, and as a result, they produce that you know 3:1 ratio, means that dominant traits will show more than 3 times than the recessive traits. So timeline, I talked about Mendel and Mischer. They actually gave us the idea about how the traits move from the parents and the parents to the offspring, but we still really did not have idea. Like Mendel, he talked about the traits, but still we did not have any idea about the gene or allele and all those. So scientists during that time, after this period, still were working on to see that who is actually responsible for the heredity. Sutton and Bovary. Sutton, during the meiosis, he observed that during meiosis that chromosomes, daughter chromosomes, sister chromosomes, actually they get separated from each other. Bovary, he was working on sea urchin. So sea urchin, what he find out? He find out that something inside the nucleus who actually control all the developmental phases of sea urchin's life. That indicates the presence of some hereditary material inside the nucleus. Okay, and that from Sutton's observation, like that during meiosis, that sister chromosomes are going to separate it. Each daughter cells are going to get the same amount of and almost identical chromosome that indicates that the responsibility of chromosome in the developmental phase or in the process of heredity. Hammarlin, she was actually working on one it's an algae, it's called Acetabularia. So this plant, this algae, it has nucleus on the foot. Okay, so the plant is like that. It has a cap, okay, and it has a base, or you can say the foot. Okay, so the nucleus remains over here, and so let's say this is the cap. What she noticed, that Hammarlin, that when she cut out the foot portion, then plant cannot grow. But if the foot is there, but they take out the cap, then still plant can grow. So that means that nucleus is responsible for the growth of the plant. Basically, the nucleus is responsible for the development of that plant. McClinton, she discovered the staining process of chromosome in corn. She was working on plant corn, and she discovered so many things like the transposition, the jumping gene. It's called transposon. Okay, that's the jumping gene. We have a small portion of chromosome that actually moves around. Okay, so we have 23 pairs of chromosome, right? That transposition or jumping gene, the jumps from number 1 to number 2 to number 6 to 23, and then from they can come back to number 15. So they moves around our genome. So she discovered that. Avery MacLeod, McCarthy, and Hershey Chase, they actually they were researching to prove that DNA is responsible for the heredity. Okay, so at this moment, during this period of time, like both Hershey Chase and MacLeod, McCarthy, during that time, people came to know about the chromosome, but were very confused, like, is it protein or is it that chromosome, DNA, RNA? Who actually is responsible for the hereditary material? So they proved that DNA is the hereditary material. Watson and Crick, they finally proposed the double helix structure of DNA. Jacob and Monod proposes the existence of mRNA. We are more towards the protein synthesis. We found out the history or the secret things about the protein synthesis. Shargrove, he discovered that always adenine goes with thymine, cytosine goes with guanine. Biddle and Tatum, they talked about that one gene and one enzyme or one protein. So the definition of genes, like gene, people used to know that time that gene is the portion of chromosome, but now we know that each gene is responsible for at least one protein. So that Biddle and Tatum, they actually discovered that hypothesis. Griffiths, he worked on the very important part of our genetic research, that's the transformation experiments. We will study about these experiments. Morgan, that Columbia University, that Flydom, it's still there. So he experimented, and he and his colleagues, they experimented on fly. They actually re-established Mendel's chromosomal theory of heredity. Okay, how much appreciation Mendel got before Morgan's work. Basically, the worldwide acceptation Mendel got after Morgan's research. Okay, he actually re-established that Mendel's principle of heredity. So Griffith's experiment. Griffiths talks about the transformation. That basically he told that bacteria, they can take a portion of chromosome or genes from the environment, and they completely changed themselves morphologically. Okay, or their behavioral pattern can be changed. So that's the transformation theory of Griffiths. So what he did, he was working on Streptococcus pneumoniae, the bacteria Streptococcus pneumoniae. So that bacteria has two strains. One is S that represents smooth, and at the same time, that's the virulent form of bacteria. Another form was R, that represents rough, means basically under microscope it looks very rough surface, and but this strain of bacteria was non-virulent. Okay, so while experimenting, he noticed that um, when he injected the mouse with that R strain, that non-virulent strain, mouse actually survive. They don't get sick. But when he injected that smooth or virulent form, that virulent bacteria actually used to kill the mouse. So what he did, he actually heat killed the smooth strain. So basically he heated up the bacteria, that is strain. Okay, he applied heat, and then he injected that heat killed smooth strain, basically the S1. Then he noticed that mouse, they don't get sick, just like R1. Okay. So basically, heat actually destroyed the toxic part of that virulent strain. But surprisingly, when he mixed up the R and that heat killed S strain, so remember over here, both this is the R strain and this is the S strain, means the heat killed a virulent form. But after heat killing, it becomes non-virulent, right? So basically in both the cases, mouse survived. So when he mixed up both R and S strains together and he applied that into the mouse, surprisingly what happened, the mouse actually dies. So why it happened? Why it happened? So he found out what happened, that R strain, means the non-virulent form, they picked up the chromosome or the gene of the S forms. So these bacteria, they became inactive, but their chromosome, when that R bacteria they picked up, then these non-virulent bacteria, they converted into virulent one. Okay, and that's the transformation. That virulent, non-virulent bacteria, they completely converted into virulent form, and that's the transformation of bacteria. And now we know that the horizontal gene transfer is there, and because of this capability of bacteria, they can pick up a small portion of genes from the environment. All the bacteria, they get antibiotic resistant, right? Now we know, but during that time we really did not have any idea about that. But this is very famous experiments, transformation experiments of Griffiths. Okay, so now we know what happened, let's say that you are sick and you are the doctor gave you antibiotic, you started taking antibiotic, and doctor told you to take the antibiotic for 10 days, but after 5 days you feel like, okay, so I'm feeling good, so I'm not going to take any medications. Okay, so what will happen, uh, let's say 60% of bacteria in your body, they are dead. Okay, but the rest of the 40% bacteria who survive that antibiotic, they get antibiotic resistant. They are the resistant bacteria. Okay, so now you carry that resistant bacteria inside your body. So from your body, the whole environment get polluted or infected with that resistant bacteria, right? Like, let's say that it's in your nose. So if you touch your nose or eyes or mouth from your hand, you can infect the environment, okay? And from there, so even if one, two bacteria are there, like let's say over here on my desk, other bacteria, uh, they actually can do this horizontal gene transfer or this transformation thing. So what they can do, they actually pick up that non-resistant genes from those bacteria, and they become um, non-resistant to that specific antibiotic. And from there, from those bacteria, other bacteria become non-resistant. So that's the problem, okay, nowadays. And we came to know because of Griffith's experiment. So you can understand, like so many years ago he did that experiment. Now we can that actually explain why and how easily all these bacteria, they are becoming antibiotic resistant. What's the secret? Secret is that transformation characteristics or horizontal gene transfer characteristics of the bacteria. Oswald, Avery, MacLeod, and Maclean. So at this moment, what happened? So Griffith actually did the experiment. He told that something actually helped to transform that rough strain, means non-virulent bacteria, to convert into the virulent one. But what it is it the protein or DNA? So we really did not have an idea. So every MacLeod Maclean, they actually extend that you can say that Griffith's experiment, where Griffith stopped his experiment, they started experimenting from there. Okay, so what they did, they actually said they did few different setup of experiment, and they used different types of enzyme that actually can destroy the protein, enzyme that can destroy the DNA, or enzyme that can destroy the RNA. So basically that time, during this period of time, like when every MacLeod Maclean are working or researching, during that time their suspect was on DNA. The scientists throughout the world, they used to think either DNA is the genetic material, or the protein is the genetic material, or the RNA is the genetic material. So there were three suspects. Okay, so so basically it was a systemic analysis, you can say. So what they did to destroy the DNA, they used deoxyribonuclease enzyme. To destroy the RNA, they used ribonuclease. And to destroy the protein, they use proteases enzyme. And finally, they found out that actually DNA is the genetic material, because when they used that deoxyribonuclease enzyme to destroy DNA, then that our cells couldn't convert or transformed into a cells. But in other cases, when they tried to destroy protein and the RNA, in every cases, our cells actually converted into cells. Okay, but only when they destroyed the DNA, that our cells couldn't convert into itself. That means, and that this experiment actually proved that DNA is the genetic material. But still, people were not that much sure. Like scientists from different parts of the world still were arguing about the genetic material. Now, Martha Chase and Hershey, they finally proved that DNA is the genetic material. So what they did, they actually infected E. coli bacteria with a bacteriophage. It's a T2 bacteriophage virus, okay? And they took two sets of T2 bacteria. One set of bacteria, they actually labeled that one set of bacteria with 32 phosphorus DNA, and another sets of bacteria virus, T2 phage virus, they actually labeled those virus with 35S sulfur. Okay, so you should know the basic concept about this theory. This research is actually the DNA has phosphorus, right? So what they did, they labeled and marked all the DNA with heavy metal, okay, 32 phosphorus. So instead of regular phosphorus, they marked all the phosphorus with little heavy metal. That's a 32 phosphorus. Okay, so all this virus DNA was carrying that 32 phosphorus. Okay, and all this group, another group of virus, T2 phage virus, their protein were marked by 35S. So sulfur is the main ingredient of the protein. Okay, so that's why they used that sulfur, okay, to mark the protein. Okay, so this group of virus, their protein was marked with heavy metal, and this group of virus, their DNA was marked. Okay, and then they treat the bacteria with both sets of both groups of virus. They ultimately found out all the bacteria. So basically, when they infected the bacteria with that labeled virus, what you already studied the life cycle, how actually virus attack the bacteria, they inject their DNA inside the bacteria, and then they force bacterial cell or host cell to multiply or to make copies of the virus, right? So that Hershey Chase, they found out that all the bacteria, all the virus came out from the E. coli who were treated with 32, 35S sulfur, heavy sulfur. They were actually not carrying any 35S sulfur. But all the virus that came out from the 32 phosphorus, labeled phosphorus, they were carrying that 32 phosphorus inside their body. So from this experiment, they proved that protein is not the genetic material. Okay, DNA is the genetic material. DNA is the genetic material. That is why all these 32P level virus, they actually transferred that heavy leveled phosphorus inside their DNA or inside their body. But protein is not the genetic material, so this virus couldn't transfer its 35-base protein into the offspring. So this experiment actually proved that the DNA is the genetic material. So nucleotides, we have two nucleotides, DNA and RNA. Okay. DNA, full name of the DNA is deoxyribonucleic acids. Okay. So DNA, or you can say the nucleotide. Nucleotide are the basically the the polymer. So nucleotide contains three parts. Okay, there are three different parts. One is the nitrogenous base, 5-carbon sugar, and one phosphate group. These three parts together compose one nucleotide. Okay, so the DNA and RNA, they are actually the you can say the polynucleotide. Okay, so they are made up of the monomer, which is the nucleotide. It's like for carbohydrate, they are the polymer, right? The carbohydrate is the polymer, and if it is sugar or glucose, they are the monomer of carbohydrate. So basically, multiple sugar together, they make large carbohydrate. Exactly same way, multiple nucleotides together, they make that nucleic acids or polynucleotides. And we have two different polynucleotides. One is DNA and another one is RNA. Now, each nucleotide is made up of three different parts. One is nitrogenous base or NB, one sugar, but it has 5 carbon. Okay, it looks like this, and one phosphate group. These three together make nucleotide. Now the nucleotides are joined to one another by sugar phosphate backbone. So remember, these nucleotides, they are the monomer, right? So monomer, they are joined together by one covalent bond, and it's called sugar. Sorry, what I was talking. Okay, so no. Okay, so nucleotides, they are the monomer. So multiple nucleotides together, they make the DNA and RNA. Okay, so if we see the structure, first we will go over the structure of DNA. DNA, it has double strand. Okay, RNA is made up with only one strand. So at this moment we are going over the structure of DNA. So these three parts, how it goes, like nitrogenous base, they remains inside. Okay, and the backbones means the main structure is made up with the sugar and the phosphate. Okay, so these two together makes the backbones, and sugars, they and nitrogenous base, they remain the inside. So this is the structure of one nucleotide. You can see the double ring structure, that the nitrogenous base. This is the sugar, 5 carbon. So this is 1, 2, 3, 4, and 5 remains over here, and that's the phosphate group. Okay. Now numbering of sugar. This figure over here, it's very important. So remember when we were studying about the structure of glucose, so this is the abbreviated form, right, of the sugar. It's C6. If you see the structure, C6H12O6, so 6 carbon is there, hydrogen 12, hydrogen and 6 oxygen is there. So it's huge. It's a very large macromolecule. So we really don't want to write all these carbon, hydrogen, and oxygen. Instead, we just write only we write down O, the starting oxygen, and we omit all others. So if we go clockwise, if we move clockwise from the starting oxygen, then this is the first carbon position, this is the second carbon position, this is the third one, this is the fourth carbon position, and the fifth carbon remain outside of the ring. Okay, this is the sugar ring. Okay, so over here it's a 5 carbon instead of 6, it's a 5 carbon sugar. And this is the phosphate group. So each deoxyribonucleotide is made up of sugar called deoxyribose. Why it is deoxyribose, I will tell you later, because one oxygen is absent over here on number 2 carbon position. One oxygen is absent, and that is why it is called deoxyribose sugar. And okay, so so this is the structure of nucleotide. You can see that these are the nitrogenous base, this is the 5 carbon sugar, and this is the phosphate group. And so this is one nucleotide. So over here in this picture we have nucleotide 1, we have nucleotide 2, and we have the third nucleotide. So 3 nucleotides are here. You can see that base, sugar, phosphate, base, sugar, phosphate, base, sugar, phosphate. Okay, now so these are the monomers. Okay, and remember that 2 monomers always remain connected with each other with one covalent bond, right? So in this case, for the DNA and for the RNA, for the nucleic acid actually, that covalent bond is called phosphodiester bond. Okay, over here they make actually ester bond, so 2 ester bonds are made, so that's why it's phosphodiester bond. Okay, now what happened over here, you can see. So number 1 nucleotide, so 1 carbon position, so this is the sugar, 1 carbon position, second, third, fourth carbon position, and that's the 5th. And for this one, second nucleotide, so this is the 4th carbon position, this is the 5th, and this is the number 3. So you can see, so these nucleotides and these nucleotides, how they are attached. So third carbon position of 1 nucleotides remain attached with the 5th carbon of another nucleotides. You can see third carbon position of 1 nucleotide remain attached with the 5 carbon of another nucleotides. Okay, so for this strand, so if it goes like that, and we know that sugar and this is the phosphate, okay, and nitrogenous base remains here. So it goes like that way. Okay, so this is the main strand that sugar and phosphate make. Okay, and that's a polynucleotide strands. So over here for this strand, you can say that this strand actually goes from 5 carbon to 3 carbon, right? So over here for this strand, if we draw a line like this way, okay, so 3 carbon remains at the bottom and the 5 carbons remains on the top for this strand, and in this case we can tell, we can say that this strand runs from, so runs from 5 prime, this is called prime, 5 prime to 3 prime direction. Okay, so it runs from 5 prime to 3 prime direction. Okay, we will come to that point again, but before that, so nitrogenous base. Okay, we have 4 different type of nucleotides based on 4 different nitrogenous bases. So we have 4 different nitrogenous bases. We have adenine, guanine, cytosine, and thymine. Okay, so if I go back over here, based on what nitrogenous base is there, we can actually name that nucleotide based on that nitrogenous base. Let's say over here it has adenine, so this is adenine, then maybe this is guanine, this is cytosine. So this part, sugar and phosphate, these 2 parts always remain constant. Only changing part is the nitrogenous base, and we have 4 different nitrogenous bases, so we have 4 different type of nucleotides. Now these nitrogenous bases, based on how many rings are there, one or double ring, we have 2 different types, purine and pyrimidine. Purine has double ring structure, but pyrimidine has just single ring structure. Okay, so adenine and guanine, these 2 has double ring structure, and they are the purine. Okay, and cytosine and thymine, they have just single ring structure, and they are the pyrimidine. So cytosine, thymine, they are the pyrimidines, and purines are adenine and guanine. So DNA double helix, Watson and Crick at 1953, they actually they first described the 3-dimensional structure, double helix structure of DNA. Watson, Crick, and Rosalind Franklin, these 3 people together, they got Nobel Prize for the discovery of the double helix structure of DNA. Actually, what happened that Rosalind Franklin, she was working on X-ray crystallization, and she first found out the DNA structure. So anyway, that these 3 together actually got that Nobel Prize. It's a big conspiracy theory. I don't want to go into that. So DNA consisted of 2 strands, 2 polynucleotide strands. In this picture, you saw just 1 polynucleotide strands, right? We have, why it is polynucleotide? Because we have 3 nucleotides over here. Actually, DNA has 2 polynucleotide strands that actually wrapped into a double helix structure. Sugar phosphate backbone is on the outside. This remains inside of the strand. So basically, if this is 1 strand, and let's say another 1 strand, because it's a they wrapped around each other. So over here, so sugar, phosphate, will be outside, okay? But the nitrogenous base, they remain inside. So they are nitrogenous base here. It's a sugar phosphate nitrogenous base here. Okay, so basically all the nitrogenous bases, they remain at the middle. Okay, and it looks like staircases. Okay, and sugar phosphorus goes like this way. That's how they are arranged. So it looks like, you know, staircase, and those staircases are made by the nitrogenous bases. Now there's this nitrogenous bases. So inside, the nitrogenous bases remain inside, and both the nitrogenous bases, like from both the strands, they are inside. And so you can imagine, like, one strand is over here, another strand is over here. We know that one nucleotide phosphate over here, we worked on that we have, in between the nucleotides, we have a covalent bond, and which is called phosphodiester bond. So over here we can understand that this strand is completely strongly arranged by or attached, you can say, with that phosphodiester bond. But how these two are together? Okay, so this is one and this is another. How these two are attached with each other? So what happens? Nitrogenous, the bases, they remains inside, right? They make the staircases, as I told. So these two bases are actually remain attached with the hydrogen bonds. So in between the nitrogenous bases of the opposite strands, we have hydrogen bond that actually hold tightly these two strands together. Okay, and these nitrogenous bases, they are complementary. Complementary means adenine always pair with thymine and guanine always pair with cytosine. So basically, if it is adenine, this nitrogenous base, if it is adenine, it has to be thymine over here. Okay, if it is guanine, then it has to be cytosine over here. So adenine always goes with thymine, cytosine always goes with guanine. Not only that, in between adenine and thymine, we have two hydrogen bond. Okay, in between cytosine and guanine, we have 3 hydrogen bond. These are the rules. Now these two strands, they possess anti-parallel polarity. Okay, so why it is anti-parallel? So take, let me change the pen color. So this is one strand and this is one strand. Okay, so you can see over here, so base sugar phosphate. So this is one nucleotide, this is second, third, and fourth nucleotide. Okay, now another rule I forgot to talk about, that every time you really need to remember that base always remain attached with one carbon of sugar. Okay, look, one carbon remain attached with the nitrogenous base, one carbon with nitrogenous base. Now always that's actually very important. And phosphate always remains with the 5 carbon. So these are 2 rules. Okay, so nitrogenous base always will be attached with 1 carbon position. Phosphate remains always attached with the 5 carbon position, and this is fixed. If you remember that, then it would be easy to understand. Now just let me, I'm looking for a space to write. Okay, so over here let's say this is 1 strand. Okay, this is sugar, so 1 carbon position nitrogenous base. This is 1 carbon. For here, this is 1 carbon position nitrogenous base. Now, and phosphate remains outside. That's the rules, right? So another strand. So if this is the sugar, this is the sugar. Okay. Same way, then phosphate should be this side, and nitrogenous base should be that side, right? It should go exactly so exactly like same way, this way. Now what happened then? What will happen? One base should remain attached to the phosphate, but actually that does not happen, right? Instead, what happens? One nitrogenous base remain attached with nitrogenous base of another strand. How it is possible to do that? What happened? One strand runs from 5 prime to 3 prime direction, and another strand is actually goes in completely opposite direction. It runs from 3 prime to 5 prime direction. Okay, so over here, so let's say this is the top part, this is head, and if this is tail. Okay, and if this is head and this is tail, instead of placing it this way, if we place this strand completely opposite way, so tail is here and head is over there, then what will happen? Okay, so we just make the strand upside down. So this is tail part. Tail goes over here instead of at the bottom, tail is on the top. Okay, so what will happen? So everything, this side will changes. So in that case, the nitrogenous base will come this side, right? And phosphate remain other side, and that happens over here. You can see. So this is the sugar 1 position base, 5 position phosphate. So this goes like that way, and that's why that 5 prime, this position is at top and the 3 remains at the bottom. So we can say that this strand runs from 5 prime to 3 prime direction. But over here, it's actually upside down. Instead of tail at the bottom, we actually changed, we get the tail at the top. So as a result, what happened? Look, so one was here. Okay, instead 1 here, 2, 3, 4, this is the 5, this is the phosphate, and this is the nitrogenous base, because we have to bring the nitrogenous base this side. You can understand, if it remains in same direction, then we will exactly get the similar structure over here, but it's not the similar. What we have to do, we have to bring the nitrogenous base inside. Okay, so that's the reason this strand goes from 3 prime, you can see that 3 is at top and 5 is at bottom. So these 2 strands runs in opposite direction, and that is why we can tell that DNA strands are anti-parallel to each other because they run in opposite directions. One goes from 5 prime to 3 prime, another goes from 3 prime to 5 prime. Over here, it's clear. You can see that this was number one position, because we have to bring the nitrogenous pieces together so that we can make one hydrogen bond in between, and that actually will help these two strands to remain together. Okay, so this strand actually upside down. Now the vertical gene transfer and horizontal. So how actually we get DNA? So there are two ways. One is from the vertical gene transfer means when we get DNA from parent to offspring. Okay. But for the horizontal gene transfer, it comes from the environment. Okay, it's actually other than this transfer. It's not from the parent to offspring transfer. Okay, bacteria they pick up chromosome or genes from other bacteria from the environment actually. So that was actually Griffith's transformation experiment I talked about. That's the horizontal gene transfer. So RNA, it's little different than DNA. Again, it's another nucleic acid, means it's a polymer and made up with nucleotide, who are the monomer actually. So RNA, it has single strand, just one strand. Okay, uses sugar ribose instead of deoxyribose. You can see over here, this is the DNA sugar and this is the RNA sugar. Okay, so DNA sugar over here, deoxy means one oxygen is missing over here. You can see for the ribose, there is one extra oxygen at number 2 position. So you can understand, right, that oxygen, so this is the number 1 carbon, this is the second, this is the third, this is the fourth, and this is the 5 carbon position. So oxygen over here for RNA at number 2 position, there is one extra sugar, and that is lacking. That sugar is that oxygen is lacking for the DNA. So that is why the name of the sugar for the DNA is deoxyribose sugar, and it is ribose sugar. So that's another difference between DNA and RNA. Now, another one, for DNA, we have adenine, guanine, cytosine, and thymine. These are the 4 nitrogenous bases. But for RNA, instead of thymine, it carry uracil. So we don't get thymine over here. In this case, we get adenine, guanine, cytosine, and uracil is actually replace the thymine. So these are the three main differences between DNA and RNA. RNA, there are 3 types of RNA are there. One is messenger RNA, transfer RNA or tRNA, and ribosomal RNA. Messenger RNA, they carries information specifying amino acid sequences of proteins from DNA to ribosome. So messenger RNA, it's actually identical copy of DNA, but it's single-stranded. So basically what happened, so if this is the cell and let's say this is the nucleus, so DNA remains over here, right? This is the DNA. So let's say this is, oh my God, cannot see. So this is the DNA. Okay. Now, so this is the DNA. DNA remains inside the nucleus, and DNA carry all the genetic information, all the hereditary information. Okay. Now the ribosome, the place of protein synthesis, that ribosome remains outside. This is the ribosome. Okay. So this is the cytoplasm and this is the nucleus. Now, when we make protein, we follow the information inside the DNA. Like let's say my eyes, it's hazel, okay? So that information is inside my chromosome, DNA, and body has to make the protein to give that color inside my eye. But the protein is going to make, this is the site, that's the ribosome, okay? And ribosome remain inside the cytoplasm. To make protein, to get that information or to transfer, to show that information, DNA has to come outside of the nucleus, right? And we actually preserve DNA very carefully because that's the only storage place of our all the genetic hereditary information. So we want to make sure the DNA is safe. So what cell actually does, cell makes a copy of DNA. Okay, so from DNA it makes a RNA copy. So this is one RNA, and that, okay, this is the RNA, and that is actually mRNA. So this is the messenger RNA. So that mRNA comes out of the nucleus and it sits over here at the ribosome, and ribosome can get all the information from that messenger RNA. So that's the function of messenger RNA. It carries the exact information that DNA carries. Transfer RNA, tRNA. So to make protein, we must have supply of adenine, guanine, cytosine, thymine, all the nitrogenous bases, right? The tRNA actually carry all those materials needed for the protein synthesis. So they actually carry lots of supplies for the protein synthesis. And ribosomal RNA or rRNA, they along with the other proteins, they make the ribosome. Okay, so over here you can see, so this is the messenger RNA, okay? This is mRNA that actually carry the exact information which was on the DNA, okay? tRNA, they brings the amino acid that needed for the protein synthesis. And ribosomal RNA, they make the ribosome. This is the, I got this table from your book. So the differences between mRNA, among mRNA, rRNA and tRNA. Please make sure to go over that. A few definitions over here. So heredity is the transmission of the traits from one generation to the next generation. Okay, from parents to offspring, how traits move, that is actually the heredity. Genetics is the scientific study of the heredity. Gene, that's the segments of the DNA molecule that actually make or responsible to make one protein. Phenotype is the appearance or expression of a trait, like what we can see from the outside, like the eye color. that's the phenotype. I'm tall or short, skin color, hair color. So all those are phenotype. What you can see from the outside means the trait that is actually going to expressed. Remember that dominant and recessive two different traits are there. So which one is going to be expressed? You only can see that traits. Okay, so basically the phenotype and genotype. So phenotype, the external characteristics or external trait that you can see. Genotype, that actually inside your body, that make up your genome. So the genotype is the genetic makeup of a trait. So one tall plant that could be pure, completely pure, means it can carry two dominant or two recessive trait, right? Or it could be hybrid. Okay, still it's tall. Okay, but it is carrying two different traits, and that is why it is hybrid, right? So that's the genotype. But the phenotype, so this is also tall, this is also tall. From outside, we cannot differentiate between these two, okay? But we can differentiate between this one and this one, because this is short and this is tall. So that's the difference between phenotype and genotype. Chromosomes are the structure that contain the genetic material. Each chromosome contains a single large double helix of DNA molecule. Okay, they are complexes of DNA and proteins. Usually, most of the time, so basically, what's the difference between DNA and chromosome? So when they're actually same, but only difference is when DNA remains associated with proteins, then that is actually the chromosome. Okay, basically the chromosome is made up of DNA and the protein. Eukaryotes, we can find the chromosome inside the nucleus, prokaryote in the nucleoid region. The genome comprises all the genetic materials that an organism possesses. Okay, so all the chromosomes you carry, like for us, we have 23 pairs of chromosomes, right? So that's not only, plus we have mitochondrial chromosome. Mitochondria, they have separate chromosome, right? So that also makes our genome. So basically there are a few diseases like that basically the mitochondria, you get it from your mom. Okay, that specifically comes from the mom. And nowadays we found out that mitochondrial chromosome carry few genes that are responsible for very, hereditary disease. So basically, that disease you get from your maternal side. Okay, so your maternal uncle had color blindness, then you are supposed to get that genes from your mom, not from your dad. So if you also have that color blindness, then you actually got that from your mom's side. Okay, so that mitochondria, that chromosome also part of our genome. So genome means total count of chromosome. Okay, the main hereditary chromosome plus the mitochondrial. For bacteria, they have one single circular chromosome, plus they have lots of plasmids. Remember that small circular double-stranded chromosome and multiple Okay, very small in short in length, but multiple plasmids are there, and they are very, very important part of bacterial life cycle. So, so together, the plasmid and one circular chromosome, they constitute the genome of bacteria. Inside the chromosome, so basically the gene is the part of a chromosome, right? So how long the gene is, that depends that how much portion requires to make one protein, that determines the length of the gene. Okay, but not necessarily all the genes inside a chromosome they can make protein. Okay, so if the genes or the portion of chromosome who can make protein, they are called gene. Portion of chromosome who cannot make protein, they are called the non-coding portion of DNA. So coding part and non-coding part. Coding, the word coding means they actually code for a specific protein. Okay, maybe for eye color, maybe for hair color, maybe for the height. Okay, so if they code for a specific protein, then they are the gene. If the portion of chromosome that does not code for any specific protein, then they are the non-coding sequence. So inside a chromosome, both coding sequence and non-coding sequences are there. Diploid eukaryotic body cells contain two sets of chromosomes, one from each parent, right? So when for each character, basically for each character, we get one set of chromosome from one parent and the other sets of chromosome from other parent. Okay, so that's how we have diploid number, means double number of chromosomes. Chromosomes in eukaryotes are located in the nucleus. So we have chromosomes that is actually 6 feet tall, so 6 feet long, okay? And you can understand the one cell, very small. So that 6 feet long chromosome needs to fit inside the cells. So to do that, the chromosome must be highly compacted and twisted, as they're highly coiled and very condensed structure so that they can fit inside the nucleus. This character is called supercoiling character. And there are lots of different types of proteins that actually help chromosome to remain as a supercoiled structure. DNA and protein complex is called as the chromatin material. Now, underroot supercoiling results from less than one turn of the helix per 10 base pair. So you can understand that DNA has double helix structure. Okay, now so when the whole thing coils within less than 10 base pairs, so like let's say from here to here we have 20 base pair. Okay, from here to here we have 20 base pair. So from here to here, within this length of chromosome, if they coiled twice, okay, then they are overwound. If they coiled just only one time, then they are the underwood. So supercoiling results from less than one turn within 10 base pair, then that's the underwood, but more than one turn within 10 base pair, that's the environment. And topoisomerase enzyme, they actually helped, or they are responsible to maintain that supercoil structure of chromosome. Mitochondrial chromosome, I already talked, exactly same way plant has chloroplast. That's the extra chromosome part. The plant, you know, and together that plant's DNA plus that chloroplast chromosome together make the genome, plant genome. Like for us, that mitochondrial chromosome plus our hereditary chromosome, the like 23 pair of chromosome together makes our genome. For the bacteria, they have plasmid, small, circular, carry few non-essential genes, but they carry few very essential, like they can carry genes for their virulence factor, for antibiotic resistance genes, use of unusual nutrients. So for the bacterial survival, these plasmids are very important. Like let's say this is a plasmid, okay? It's a circular and double-stranded chromosome, okay? There one starting point should be there from where plasmid can start replicating. Anyway, so let's say inside this plasmid, they have one gene, let's say it is penicillin resistant gene, okay? Now, so basically, if this gene is present inside the plasmid, means genes means it actually represents a protein. So which protein it is going to make? It is going to make protein that is anti-penicillin, right? So basically, if this protein is present inside that cell, then that can destroy the penicillin. So basically, by carrying these genes, penicillin resistant genes, this plasmid can help bacteria to overcome the action of penicillin. So the bacteria with plasmid, which is carrying the penicillin resistant genes, it is very difficult to destroy. You cannot kill that bacteria with that antibiotic or the penicillin. Okay, so that is very important. And you know what, that plasmid, they frequently, bacteria can pick up this plasmid from the environment very frequently. So if this is one area, multiple bacteria are there, okay, and they can actually release their plasmid in the environment, and one bacteria can pick up another one's plasmid. And let's say this bacteria was not carrying any penicillin or antibiotic resistant genes, but once it pick up this plasmid, this bacteria will convert into another strain of bacteria, which will be penicillin resistant. And that's the transformation, or you can say that horizontal gene transfer. And that is why this plasmid is very important for the bacterial survival. Okay, their own circular chromosome does not carry this antibiotic resistant genes. Bacteria actually get that antibiotic resistant genes from the plasmid. They carry these genes inside the plasmid. So plasmid is very important for bacterial survival. Okay, we are done. Thank you. If you have any question, shoot me email.